Hey everyone, it's Jamie here, dropping in before the episode to give you two exciting updates on In Plain English. The first is that In Plain English is going to be having our very first live show next week on Wednesday, June 12th, at the University of Pennsylvania in Philadelphia, Pennsylvania. This live show is going to be focused on talking about rigor in science. What is it? How we can improve and increase the rigor of scientific studies, and basically what we can do to change the culture of science. This panel is going to be co-hosted by the Community for Rigor, who I've been working with to put together this event and who initially suggested the event. So I'm super excited to be working with them. The other panelists will be Conrad Cording from Community for Rigor, along with Kayla Singleton, the founder of the Black and Neuro nonprofit, and Arjun Raj, a professor at UPenn and a champion of scientific rigor in his own lab and writings. If you are in Philadelphia or can get to Philadelphia for June 12th, you can sign up to attend the live show. It's totally free at the link in the description, or go to the website and you can see the flyer and scan the QR code. If you're not going to be in Philadelphia, don't worry. This live show will be recorded and then uploaded as a regular podcast episode in the In Plain English feed at the beginning of July. So you will still get to hear this conversation just a little bit later. The second exciting update is that, as you may have noticed, In Plain English has a new logo. I am so excited for this logo. I think it looks excellent. I think it really pops. And it was designed by three of my wonderful friends and colleagues, Jessica Higginbotham, Tania Lintz, and Yolanda Campos. The three of them have started a scientific design company called Biographics. So if you are looking for some cool designs for your research project or your science communication project, you can find their information in the description and check them out. With all that out of the way, let's get to the episode. Hello, everybody, and welcome to another episode of In Plain English. I am your host, Jamie Moffa, and today we are going to be talking about a immunology paper called Antigen-Specific Depletion of CD4 Positive T Cells by CAR T Cells Reveals Distinct Roles of Higher and Lower Affinity TCRs During Immunity by J.U. Yi et al. And here to explain what all of that means is our expert, Clarice Pavlak. Clarice, would you like to introduce yourself?
Absolutely. So my name is Clarice, and I'm in my second year of my PhD at Washington University in St. Louis, and I'm in the lab of Dr. Gregory Wu, and we focus mostly on an autoimmune disease called multiple sclerosis.
Great. And thank you so much for being on the podcast. Our two guests for this episode are Johari Hunt and Andrew Holder. So would you each like to briefly introduce yourselves?
Sure. Yeah. My name is Johari Hunt. Some folks may call me Knight or know me as Knight, like the time of day, and that's completely fine. I use he, him pronouns. A little bit about me. I am a SoCal baby, born and raised, but I currently reside in St. Louis, Missouri, and I'm really loving it out here. We get snow, and I know other people get snow in California, but I didn't. I am by trade a religious professional, and I currently work for First Congregational Church of Webster Groves, which is a UCC denomination.
Great. Thanks for joining us.
And then I'm Andrew Holder. I've born and raised in St. Louis, and I'm currently a second-year law student at St. Louis University, pursuing a concentration in health law.
Awesome. Well, without further ado, let's start talking about this paper.
All right, sounds good. So, as Jamie introduced, this is a paper about this CAR T-cell therapy, and sort of what the authors are doing is they're taking this sort of novel live cell-based therapy design that's been used in the context of cancer and like treating cancer, and they're interested in bringing it into the world of autoimmune disease as a potential treatment. And sort of while they're doing that, they're also thinking about how can we use this therapy as a tool to sort of understand more about the mechanisms behind autoimmunity. How this CAR T-cell therapy works is there's a type of the immune system is focused on eliminating germs and protecting us from foreign invaders. And so kind of in the context of cancer, the immune system is also able to fight cancer cells. And so scientists have been really interested in how can we better equip the immune system to fight cancer cells. And so there's this one kind of immune cell called a T cell, and specifically cytotoxic T cells. What they're able to do is actually sense whether there's something wrong or disturbed with our body's healthy cells, and they can then kill cells that they identify as being cancerous or virally infected and whatnot. And so for this CAR T cell, which is called a chimeric antigen receptor T cell, scientists will collect T cells from a cancer patient, and they can modify them to better target and kill the cancer cells, and then take these engineered T cells, return them back into the patient, and then they can begin killing the cancerous cells within the patient. And so sort of there's a similar idea in the context of autoimmune disease. So autoimmune disease is when the immune system is supposed to attack foreign invaders, but instead it starts attacking the body's own healthy tissues as if they are foreign invaders. And so by engineering the immune cells, the goal is to be able to target and kill the immune cells that are driving this self-immune response that's damaging the body's tissues.
Okay, wait, I'll jump in real quick. So cancer, right? What I know about cancer is that it's just a cell. It's any cell, right? Like it could be a liver cell or a lung cell, or something ends up where it's not supposed to be, and then it starts reproducing itself, damaging the healthy tissues around it, right?
Yes.
So I imagine, and then I'm intimately familiar with autoimmune disorders because my biological mother has lupus and fibro, and so I grew up around it. And so what I'm curious in your explanation, right, is how are the T cells, because I imagine in cancer research, this would be aimed like at a specific area of the body, I guess, or maybe it's some medicine, but it can be trained to target specific cells. So for the autoimmune cells, how does it know which ones are the ones that are causing the specific response?
Yeah, that's a great question. And so right now, identifying what these differences are between like healthy cells, cancer cells, autoreactive cells is sort of a big field of where CAR T is going. So right now, the current CAR T cell therapies that are on the market and being used to treat patients, they're targeting a specific type of immune cell called B cells specifically. And so because B cells, you can live without them, these therapies are able to specifically target a molecule that's unique to the surface of B cells, and it depletes all of the patient's B cells in total. So it's eliminating cancers that arise from B cells by just depleting all of them.
I see.
And so that's kind of where like the current CAR T cell therapies stand. But in the context of autoimmune disease, so the T cells or the cells of interest that we're interested in depleting are specifically T cells that are driving autoimmune disease. And so sort of what T cells do is they survey the host's healthy tissue or healthy cells, and they look for cells that have become damaged in some way. And the way that they do that is all the cells in our body display this molecule called the major histocompatibility complex, or MHC for short. And I like to think of MHC sort of as X-ray vision, where it'll present bits of proteins that are inside the cell. So that way it's visible to the T cells, and they can determine whether these protein bits are from normal proteins within the cell, or they could be from a virus, or they could have become mutated in a cancer cell. And it can kind of sense the internal state of the cell, whether it's healthy or there's some sort of process going on that shouldn't be there. And so when you want to deplete a T cell using an modified T cell, what they're doing is actually taking these MHC molecules and putting a peptide of interest on them. So it's kind of acting as like a bait that will lure the autoreactive T cells by putting their target on the surface. Mm-hmm. So like in the context of this paper, they're studying a model called experimental autoimmune encephalomyelitis. And this is a model that's meant to represent multiple sclerosis, which is an autoimmune disease of the central nervous system. So meaning the brain and the spinal cord are affected. And specifically in multiple sclerosis, we don't actually know what the self protein, or we would refer to it as an antigen, that's driving the disease, but we suspect that it's proteins in the myelin sheath, which is what insulates brain cells so they can send messages very quickly and efficiently. And so the thought is that the immune system is recognizing these myelin cells and it's damaging them, and that's what's causing multiple sclerosis and all the symptoms that come from it. And so we have an animal model that's based on one of these myelin proteins called MOG, where actually if you take a mouse and you give them essentially like a vaccine we call immunizing, where you have the myelin protein and then you put adjuvants that kind of help to just stimulate the immune system non-specifically, it causes this immune response against MOG, and then results in the immune cells attacking the myelin sheath that has a lot of MOG expressed on the surface. And so by taking these MOG peptides, or bits of proteins that we know are being affected, you can put those on these like MHC molecules on the CAR T cell, and that will attract only the T cells that can sense and respond to MOG.
So I guess my knowledge about like science and like cells comes from like almost like a high school like standpoint, like not really taking many like science classes even in college, and like just kind of watching like John Green videos to get by, where everything's a little like animated. And so I guess I've never necessarily thought of cells as being smart and being able to like identify this stuff. And so I guess like what about like the T cell makes it different in this way, that it can like go and it can like identify these cells that have these like proteins or these peptides in it and then respond to that?
Okay, I think that's definitely a great question to start with some more of the basic immunology here. So I guess I'll start with the immune system has two major branches. There's the innate immune system and the adaptive immune system. And so the innate immune system has predetermined receptors, which will sense things in the environment. And so they've been through like rounds of evolution, they've been tailored to specifically target like different features of bacteria or viruses or germs, whatever, foreign invaders might be trying to like infect the body, versus the adaptive immune system really tries the goal of it is just be able to target any kind of biological molecule that it may encounter. And it does this by actually randomly creating receptors for each immune cell. And so this kind of comes with the pros and cons, where because the receptors are being randomly generated, each receptor there's like maybe like 10,000 different specificities, or probably more than, it has to be more than that, 10 to the 7th, like 10 million different specificities among like all of the immune cells that you would have in your body at a given time. So it's really this like random generation of the receptor. That's what's giving each immune cell or each adaptive immune cell its ability to like see something specific. So then when you kind of consider this collection of all the different immune cells or adaptive immune cells that you have in your body at one time, they can see all of these different potential molecules that they might encounter. So kind of when I refer to like a T cell that's able to see MOG, it's because it's randomly generated receptor that it happens to recognize that structure, compared to like there might be most of the other T cells in the body are going to recognize a different kind of molecule structure.
Are these T cells like allocated to specific parts of the body then, or are these like MHC cells that are found in these like immune system disorders like found in certain parts of the body? Because I know, like as Knight mentioned earlier, like with something like lupus, like it does like primarily affect like certain parts of your body, like I believe like the chest area as well as like arms and like areas like that. So are the MHC cells like specific to these areas, or are they found everywhere?
So MHC is on every cell that's within your body, but kind of like what you're getting at, there's different tissues that have different functions. So depending on what kind of cell the MHC molecule is on, it's going to display different protein fragments based on like what it has inside of it and what it's made of. And so what T cells can do is they don't necessarily like know what their target is when they exist. They will actually circulate through your blood, and then they can go into your different lymph nodes, which are like these little structures that you have kind of all over your body. And so what they do is they drain fluid from the tissues, and that will go into the lymph nodes. So if you have, let's say, like an infection somewhere, then bits of the germs can kind of drain into the lymph node, and the T cells will circle through and they can scan the lymph node and see if they detect their antigen, or what we call their target. So T cells kind of don't know necessarily like what tissue they would respond to, but they circulate through the body and are able to like find it by surveying sort of the substances that are being drained into the lymph node at a particular site.
Gotcha.
So I have a quick question. Can you define MOG again? I don't know if you already said it and it got lost in translation, but can you be a bit specific, because you keep saying that So I want to make sure I have a correct understanding.
So starting with the central nervous system is composed of the brain and spinal cord. And so brain cells are like the individual units that make these up. And brain cells are surrounded by what's called the myelin sheath. Mm-hmm. And pretty much what the myelin sheath does is it just insulates the brain cells so they can send electrical signals very fast and efficiently. And then the myelin sheath is composed of a variety of different proteins, with MOG being one of the proteins that's on the surface of the myelin sheath.
Okay.
It gets targeted, degrades the insulation, and then kind of like if you have, uninsulated wires running through your house, it causes problems.
So one of my questions is, I'm a humanities major. So just what kind of citation do like science papers use? Like when I was looking at, like when I read through your paper and then I was like looking at your citations, I was like, where do you cite this when you're reading the paper? I was just wondering like how citations particularly work here. And then my other like more technical question to the paper is, how do you know which tests to run? Like, is it like y'all kind of already have an idea and like this is part of your lab and you're publishing on kind of like what's going on in your lab? Because like with your charts, right? I'm like reading the description of your charts, like this is a very precise thing you're talking about happening and observing and hypothesizing and then declaring the results. So I was just wondering like how do you find the specific methods kind of to precisely use to measure what you expect the MHC cells and the CAR-D4 T cells?
Okay, so for citations, so it kind of depends a little bit on the journal that you publish in and what their specific requirements are. But usually you'll have some sort of like in-text reference. And then at the end, you have like your reference page and it'll refer to specific papers that have been done. Mm-hmm. So usually like the references are all to like other scientific papers. And it's either like you're citing like a finding from them, or you might be citing like a method or something like that.
I was going to say like the actual in-text citations in this paper are the numbers in parentheses. And then you can go, if you go all the way to the back several pages, there will be a number next to the name of another article.
Mm-hmm. Yeah. Okay. It's kind of like footnotes, which I thought, but I wanted to make sure I was understanding the references correctly.
Okay. So then your second question about methodology, that's a really interesting, like fundamental question of science is how do you know like what techniques and stuff to use? So I guess sort of the process of how that happens is you think the first step that happens is you have some sort of question that you're asking. So some sort of like scientific idea. And then you're like, okay, this is what I think might happen. And then you start to think about like, well, how can I address this question or figure out like my hypothesis? And so I guess in terms of methods, like kind of thinking about in the context of this paper. So I think kind of like the person who's kind of the lead scientist on this paper has an expertise sort of within this realm. And so I think they're kind of familiar with a lot of the methods that other people have been used. So kind of a really important like task as a scientist is just reading literature and kind of learning what's been done in the field. And by reading other people's work and other people's findings, you can also see what methods they were using and how kind of the context of when you would use that method and what's appropriate. Mm-hmm. So that's kind of an important concept of learning kind of what are the methods that are generally used in my field of work. And then depending on what your question is, you can kind of tailor that method to what you're trying to accomplish.
Okay.
So there's a sentence on page 10 that I was looking at that I wanted to ask you about, where it says higher affinity autoreactive T cells provide initial activation energy to initiate autoimmunity, analogous to the energy required to initiate a chemical reaction. And so for that sentence, I was wondering, I know autoimmune disorders are extremely hard to spot and diagnose, especially after they've happened. But reading that sentence almost suggests it's would be easy to spot this almost preponderance of auto or of T cells that exist within the body. So I guess I was just kind of wondering what that sentence meant in this context.
So I guess in this context, that's kind of their interpretation of the experiments that they did in this paper and sort of what they think or what their kind of model of how an autoimmune disease happens based on their findings, because autoimmune disease is a very complex topic where in a lot of cases it's very unclear how it's being initiated, and it's not really sure. There's a lot of theories, like there's genetic components where some people have certain features about them that put them at risk for developing it. And then there's also another really important component where there's thought to be some sort of environmental trigger that then is launching the immune system to start attacking its own tissues. And it's kind of thought that, I guess some examples could be like a viral infection might sort of trigger some cells to start attacking the body's tissues. And a couple ways that could happen is like what we call bystander activation, where the immune system's trying to fight the virus, but for some reason, these other cells that are reactive to the body kind of accidentally become activated just because there's so much inflammation and such a strong like immune response ongoing. Or molecular mimicry is another concept or idea of how autoimmune diseases might be triggered, where your body's trying to fight a virus, or some sort of germ, and then part of the germ also looks like one of the molecules that's within the body. So the immune cells that are attacking the virus might also accidentally attack self-tissue because they look similar enough. So that's kind of like some ideas that we have currently about how autoimmune disease is starting. But beyond that, we really don't know what the actual mechanisms are of why these self-immune responses are occurring, because we don't want them to.
I think if I can jump in from a clinical standpoint, diagnosing autoimmune diseases is also difficult because the clinical presentation is very vague. So a lot of times what you'll find with autoimmune diseases is that the symptoms are like fatigue, pain, like generalized not feeling good. And until it progresses to a like later stage, those symptoms might not become more focal into something that's easily recognizable as like this autoimmune disease. So oftentimes you go through a lot of other tests and diagnoses for things that are more acute, right? Because also doctors want to test first, like, do you have cancer that's going to kill you like soon? And then only once you like rule out all the other options will a lot of doctors be like, okay, maybe we should look into autoimmunity. I don't know. Knight, was that like kind of you said your mom has a couple of autoimmune diseases. Was that like her experience?
Kind of. My mom had a very, it was so much, they didn't believe that she had it for a number of years. So it wasn't actually until a nurse had recognized her symptoms and then asked the doctor to perform a specific test were they then able to actually give her a diagnosis for lupus.
Yeah. That's also a very unfortunately common like occurrence in medicine, is that people don't get believed when they say that they have something, or that when they say that they're concerned about something, and that can lead to really long delays in treatment. There's a lot of like reasons that are not just whether or not it's easy to test for an autoimmune disease, because you have to know kind of like how you have to know what kind of scientific test to run. You have to know what test to run to catch this diagnosis.
Yeah.
I think a lot of that too is we don't really have good diagnostic tests because we don't really understand what's happening in a lot of these diseases. So like this experimental autoimmune encephalomyelitis model, like we know that MOG is the target and it's very clearly defined. But in multiple sclerosis, we don't actually even really know what the actual targets of the immune cells are. So that's kind of one limitation that we have, is like we don't really know what they're attacking. And within a human system, like it's harder to address those questions, and kind of because we can't perfectly recapitulate multiple sclerosis in a mouse. So that's kind of where some of our limitations are coming, and why we're not quite like there yet. And that is sort of a big emerging topic in the field, is trying to better understand like what are the targets of these immune cells in these different diseases, and kind of hoping that can lead to better diagnostic tools and better therapeutics. So we still kind of have a lot of limitations surrounding that.
And so I guess from like a clinical standpoint, what is the test? Is it like kind of symptom based, or does it like involve stuff like what was in these papers, like looking at T cells and like looking at stuff like this and running labs? I guess I'm sorry, I feel like I don't know a lot about like autoimmune diseases and like conditions, and so I'm just trying to gain almost as much context as possible.
Yeah, no, that's a really good question, because in the clinic, so there really aren't that many tests or screening that you can do that's like available for a lot of autoimmune diseases. Kind of like what Jamie was getting at, is there's sort of a lot of different vague symptoms that a person can have. So what's really important is this evaluation of someone's clinical history, like what are the types of symptoms they're experiencing, how did these symptoms appear, kind of like how old are they, and sort of a lot of those questions can kind of factor into it. There is some more generic testing that you can do, like for multiple sclerosis specifically, doing a lumbar puncture and looking for what's called these oligoclonal bands is an important test. And so what oligoclonal bands are is just a certain test that they do, and they can see whether there's antibodies present in your cerebral spinal fluid, which normally there shouldn't be, but in almost all multiple sclerosis patients there is. So that's sort of, even that alone is not like a super definitive test, because there's a lot of other diseases that can also have oligoclonal bands present. Something else that they'll do is MRI findings is also really important, because in these areas that you get where the immune system is attacking the myelin sheath and it's getting damaged, you can see lesions in the brain, which are like little scars where the myelin's being damaged and the immune cells are all infiltrating that normally wouldn't be there. So kind of based on patterns of these lesions that they're seeing in the brain, that can also help contribute to a diagnosis. So kind of with a lot of autoimmune diseases, you really have to consider this big picture of results from various different tests that we have available, as well as sort of the clinical symptoms and clinical onset of what they're experiencing.
So since we're talking about clinicals, what is hypothesized as like the ideal body or standard or like baseline that I guess this research is aiming for? Because I'm also thinking about, right, is like who, like when we think about autoimmune diseases and especially like what causes flare-ups, like environmental factors are huge. Like, you know, stress will kill you even without an autoimmune disease. Just thinking about like certain environmental factors that would affect making this like clinically possible, and like thinking about if it is going to deplete the correct T cell, like how you know it's working like the same and like a white person versus a black person. Like ideally it's like, well, we're all the same until we're not, you know?
Yeah, no, you bring on a good point that autoimmune diseases are like very individualized. So it is kind of hard. We don't really know what's going on in each individual person. Mm-hmm. And there is like a lot of difference. So I guess kind of the end goal for these therapies is someone who has like whatever XYZ symptoms for their disease is that they don't have anything anymore. So no symptoms. And also if they had certain positive clinical tests, that those return to what you would see in a baseline. So those are kind of the big generic outcomes. But I guess like what you're saying with different like racial groups or like sex difference is really big in autoimmunity. Mm-hmm. I guess like there are definitely, I know there are people who are doing specific studies on that, like differences between groups and if there's differences in disease mechanisms. Mm-hmm. But I guess for now, we don't really know kind of the differences. And you see that with a lot of the therapies that are available. Like I'll use an example, is this one therapy called adalimumab. You might have heard of it as Humira. Mm-hmm. And it's an antibody that's targeting a signaling molecule called tumor necrosis factor. And it's a really, really beneficial drug for people with inflammatory bowel disease, rheumatoid arthritis, and several other autoimmune diseases. And it only works in about 50% of patients, but we don't really know why. So I think there's kind of this emerging theme that the more we study the mechanisms of disease, the more we're starting to see like further subclassification of people being broken down into different diseases. And actually a good example of this is the one that I studied. So multiple sclerosis is kind of the umbrella of these autoimmune diseases targeting the central nervous system. But like more recently, this rare disease called MOG antibody disease, where it presents similar to multiple sclerosis, but they found that there's actually subtle differences in what symptoms, like people tend to get more visual problems and problems related to inflammation in the spinal cord. So like lower limb paralysis or weakness and like bowel incontinence and stuff. And so these diseases kind of used to be lumped together. But when people started to do more testing and realize some subtle differences in like having antibodies against MOG in the serum versus not, where it kind of became two separate disease entities. And so I think there's kind of, to your point, there's going to be a trend moving towards that, where I think the more we learn about these diseases and the more personalized it gets, the more like subclassing we're going to see. And then that will allow us to make therapies that are more targeted to specific diseases. And another thing that's kind of important to note too about like MOGAD or MOG antibody disease versus MS is that the treatments for them are different. So it is important to know which one you have, because that'll allow the patients to get to a therapy so that works for them like more quickly.
I think another cool thing about the CAR T cell technology is that it really is a very personalized therapy, because they're taking your T cells, right, and then modifying them and putting them back in you.
And that brings up a good point too. Like I had kind of mentioned that like in multiple sclerosis, we don't really know what exactly the targets are. So I guess kind of when CAR T cells, or when they're kind of going into practice in like actually using them to treat patients, I envision in the future that there's probably going to have to be like some upstream testing that would have to go on in a patient first before you could use CAR T cell in, where you can kind of identify like in this specific person, what are the antigen targets that the immune cells are really going after. And then you could tailor the CAR T cell therapy to go after the immune cells with those specific targets. So that's kind of I guess like a limitation in sort of the CAR T cell therapy. At the current state is like in this paper, they're assuming that MOG is the target, but that might not be the case in like all instances of MS. So this specific therapy that they're kind of looking at might not apply to everybody. On
page 3, when they're talking about sequencing the different alleles in order to get the, I'm assuming the MHC to work with the CAR T cells, how do you think that sequencing is going to look? Like, well, y'all are just saying like how individualized it is. So I guess in order to make this like applicable for people, they would have to get their cells sequenced and then have the medicine introduced back to their body.
Yeah, you bring up a good point that, so the MHCs are actually the most polymorphic genes in the genome, meaning out of all the genes that exist, they have the most variation among people. So that is kind of a contributing factor as to like what complicates autoimmunity is every person or every patient that you see, there's going to be a lot of variety in what MHC alleles that they have. So you would have to sequence your patient ahead of time and kind of design your CAR T cell therapy based on what MHC allele they have. And so kind of something a little bit more into the weeds I didn't quite get into is like, so each person has like a different set of MHC alleles, and there's a lot of variety between patients. And that also influences which protein bits that your cells are going to display. So even if you have, let's say, like 5 people with multiple sclerosis, they all have different MHC alleles. They might all have immune cells that are reactive to MOG, but each of their MHC alleles could be displaying different bits of MOG. So that is something you would have to consider when designing this CAR T cells. You would need to figure out what their MHC allele is and figure out which bit of MOG is being targeted to then go and make your CAR T cell that's displaying that MHC with that peptide and depleting those specific cells. So that is something that would have to be done upstream before the CAR T cell therapy can be developed for a specific individual. But that being said, another important thing to note is there are some specific risk alleles for MS. So certain MHC alleles are associated with like an increased risk for developing MS. And I think in this paper, they mention the DR4 allele that presents MOG 97 to 108, so that specific peptide. So by developing sort of a CAR T cell like that, it might have a little bit more universal utility among people if there's kind of a group of these patients that all share this particular risk allele for MS. So it wouldn't be like every single person is going to have their own CAR T cell developed.
Okay.
But you do have to like check their MHC type and see which one they have and decide whether this therapy would be relevant for them.
Okay.
I think maybe we should get into some of what they did so we can start talking about like how they were actually developing this potential future therapy.
Yeah, okay. So just to kind of run through, I guess, sort of the workflow of the paper is so kind of the reason for doing what they were doing stemmed from this idea is they want to learn more about the T cell response that's happening in autoimmune disease. And sort of this emerging theme is, so T cells can vary in their affinity for their peptide MHC target. And that basically means how strong their receptor can interact with the peptide MHC physically. And so it's kind of thought that, because in the context of infection, you want the T cells that have the strongest affinity for the target. Those are going to be the most potent and the most important. But sort of in the context of autoimmune disease, there's this rising idea that might not necessarily be the case. that there's evidence that a lot of these lower affinity TCRs might also be really important at driving disease, in addition to these really high affinity ones. So they were kind of developing this CAR T cell and sort of evaluating, based on which T cell populations they were eliminating, kind of what does that say about their role in disease. And so they start by developing their CAR T cell. And so how they do this is they take T cells and then they designed this artificial receptor. So they're, instead of having a T cell receptor, they're now giving it an MHC molecule with a MOG peptide in it. In this case, it's called MOG 35 to 55. That's like the bit of MOG that binds to this MHC that they're using in mice. And then they keep this some of the same signaling domains kind of inside the T cell. But basically what they're doing is they're designing their CAR T cell. So instead of having a regular T cell receptor, it has this MHC molecule that's displaying a MOG peptide in it. And sort of the premise is that this MHC with the peptide in it is going to attract the T cells that are normally attacking the myelin sheath in this mouse model. And it'll be able to deplete and kill these cells.
The job of those T cells is to kill other cells. So like it has this lure. It's like those fish, right? With like the little light lure. And so like the immune cells in the mouse, in this instance, that are reactive against this MOG antigen will go towards the light lure, and then the CAR T cell goes ow and eats it.
Nice.
Okay, so that's kind of like the premise of this cell therapy and what they're trying to do. And so pretty much they have their like therapy designed, and they do some validation to show that it's killing the target cells that it wants them to be killing. And so then kind of what the interesting starting point is they have the EAE model, the experimental autoimmune encephalitis model, and they're like, okay, in this model, what we do is we immunize mice with MOG. So you're injecting them with this MOG peptide and then giving it adjuvants to sort of just generically stimulate the immune response. And that's causing the mice to generate an immune response against MOG. And they develop hind limb paralysis, and it kind of goes through like a little bit of a relapsing disease course, somewhat similar to what you would see in multiple sclerosis. And so what they did was when these mice start developing these symptoms of this hind limb paralysis, they're injecting their CAR T cells to see what happens in terms of the disease context. And so when they do this with their initial CAR T cell that they develop, they find that it is pretty effective at stopping the disease from getting to its full severity. It doesn't completely stop it, but the disease they get is very mild compared to what the mice would get if they're not treated with the CAR T cells. And so that was kind of their first showing of, hey, we can actually use these CAR T cells to ameliorate disease in these mice, or at least in this model that we have of multiple sclerosis. And so then they're kind of thinking a little bit more about this CAR T cell, and they're like, well, how sensitive is it, and what T cells is it actually depleting? That's kind of the big question that they're interested in, is like, what is this CAR T cell actually depleting so that it's minimizing disease? And so then they kind of develop an interest of looking at what's called the 2D2, which this has been a T cell receptor that's been identified, and it's known to respond to the MOG 35 to 55 peptide. And it's known to be sort of in the class of these lower affinity TCRs. And so they're interested in, well, can our CAR actually deplete these T cells that are considered lower affinity? And when they do some testing in vivo, they find that they can't, that they're not able to deplete these lower affinity T cells. So then that prompts them to make some modifications to their CAR T cells. They make one modification that makes the peptide MHC complex on the surface more stable. So there's more of it on the surface, and it increases the sensitivity. And then they make a second modification that increases the CAR T cell survival so that they stay around and live longer when they're inside the mouse. And so then they want to see, how does this modified CAR T cell sort of fare when they put it in compared to the original one that they had. And so again, they do the same thing where they use this model where they're immunizing the mice with MOG. And then they kind of split the mice into two groups. So on day 12, they split the mice into the group of ones that have no symptoms yet and some that are starting to show symptoms. And they find that in the mice where they had no symptoms, when they start giving them the CAR T cells, they basically don't develop disease with this CAR T cell. So they were almost able to like completely prevent hind limb paralysis onsetting in these mice. And then in the ones where they did sort of have a little bit of disease starting, they found that it was able to kind of also curb the disease, and they don't really develop symptoms compared to the mice that aren't treated would go on to develop this hind limb paralysis. So they're showing that it's fairly effective at preventing disease from onset. And so that's great that they're able to stop disease. But when you sort of think about the context of a person, like, how do you know when someone's going to get multiple sclerosis? Like, you don't. You really don't until they already start having symptoms. So what they're really interested in, that's most important for a therapeutic context, is, well, how can these CAR T cells stop disease once it's started? So that's kind of really the next big question that they have. So instead of injecting the CAR T cells when the mice start to develop disease, they wait until they're sort of at their peak disease. And at day 18, they give them the CAR T cells. And here, unfortunately, it's not quite as exciting. They do see that there is a reduction in disease severity, but it's not able to completely cure them of the hind limb paralysis that they have. So kind of the takeaway is that these CAR T cells, they're able to prevent disease and they can ameliorate disease once it's started, but they're not quite as effective at that. So there's still a little bit of room for improvement here. And so then now that they kind of have shown like the efficacy or the efficiency of these CAR T cells and the fact that they are able to stop disease, they're kind of interested in, well, what's actually being depleted by these cells and like what's actually happening in the mouse, because they're seeing the clinical outcome, but they don't actually know what's being targeted, what cells are getting killed.
So you mentioned in order they for the mice, they injected it at the peak of the, I think, the flare up, right? And it was supposed to be like so many days after the mice were displaying the max symptoms. Is that like a requirement, or were they just doing it to kind of see its full potential? Like if someone's already been diagnosed with MS, for example, and if they caught it early, could they start this treatment before it got that bad in order to like prevent the damage from happening? Because I imagine once the damage is done, it's done. Like once the cells are destroyed, it can't be replaced.
Generally, like a lot of these papers, you'll kind of see they start with prevention because it's generally easier to prevent rather than to treat. Because like you said, there's a lot of like damage that's ongoing. And even when the mice recover and aren't having symptoms, there's still changes in them that happen because of like the response. So that's kind of like a tendency to go for like a preventative first. But then they do want to treat at peak disease because that's more representative of what you would see in the clinic based on our ability to diagnose these diseases now. Because kind of like we talked about earlier, where there's no real definitive test to diagnose for autoimmune diseases, it can be very tricky to rule out other diseases and get to the correct diagnosis. So a lot of times in clinic, it can take people like several years to be diagnosed. So kind of based on the landscape of our ability to diagnose now, I think there's a stronger interest in being able to develop something that you can use to treat when someone already has disease onset. So that's kind of where giving the CAR T cells at peak disease is kind of ideally you're reflecting what you would see in the clinic. Like you're saying, it would be ideal that if we could get something preventative to do that instead. So for example, in the case of diabetes, or type 1 diabetes, which is the autoimmune form, like most people that get diagnosed, they get diagnosed because they already have symptom onsets. But in the instances where people have a strong family history, they can actually detect the presence of certain antibodies in the blood of children kind of young and be able to sort of predict whether diabetes is going to onset. There's kind of like a pre-stage where they don't have symptoms, but you can kind of see some of these immune changes. So in a case like that, you really would want to treat with CAR T cells as early as you could to kind of prevent it from going on to damage the islets and produce symptoms. You really want to treat early on before that happens. So that's like an example of where with CAR T cells, you would want to treat earlier, and that would be ideal. For MS, we don't know any indication of like before someone would have onset of symptoms, even if there is like a family history. So for MS, it would be a little bit more complicated and unclear, and we would be going more for a treatment versus a preventative. But for like in the case of diabetes, you make a good point that if we could give it to people that have a known family history and they're kind of on track to develop diabetes, that would be ideal for minimizing the damage. But in humans with multiple sclerosis, there is a degree of regeneration or remyelination that occurs. A lot of times the lesions can heal and people's symptoms will go away. So they might still have like a scar that you can see on MRI, but their physical symptoms like go away because there is an extent of healing. In some of the mouse models, some of the models, there is evidence of remyelination and others there aren't. So that could be a reason why in this model, the mice, like there is some relief of their symptoms, but it doesn't go all the way away because it might not be repairing. But I think in humans, there is like the ability for the remyelination and the repair to happen.
Earlier you were mentioning how right now we're at a point where we're like specifically altering like individual T cells. And so we're taking them from the person and then we're giving them back basically after they're altered. But you also mentioned the possibility that eventually if like enough people are similar in a certain respect, like with any form of autoimmune condition, that we could almost develop like a one size fits all kind of T cell to give them. So how would that, I guess, like work logistically? Are they easy to like store, or like would they be like donatable, or like I know that's not your job to like figure out, but I guess I just wanted to hear your input on it.
Oh no, that's a good question, because I guess I should clarify what I mean by a one size fits all. So I think in the case of CAR T cells, you would always want to use that patient's T cells. But I think kind of what I was referring to is the specific construct, or like the actual chimeric receptor that you're putting into them to engineer them, that you might be able to conserve across people.
Oh, okay.
So like, for example, with the therapies that are on the market now for B cell cancers, there's a marker on B cells that's shared across everybody. So what they're doing is they take the T cells from the patient, they give them this construct that they give to any patient and then put them back. So it kind of is universal in that sense. Whereas with the MHC CARs, because everyone has different MHC alleles, you would have to like allele match to make sure they have the right one. But I was kind of getting at where like if you have this population of people that have a shared risk allele, you could use the same construct for those people because they have that same allele in common, compared to like for each person, you would have to make a new construct depending on what allele they have.
Gotcha.
So I think that's kind of more where I was getting at. But at the end of the day, you would always want to use that person's T cells to modify the therapy, or at least that's what I think based on what I know, given like with the CAR T cell field now.
Gotcha.
Yeah. So then the second kind of the paper, or second part of the paper, is more getting into what are they actually learning about the roles of these different T cells in autoimmune disease by depleting certain populations. And so what they do is they go ahead and they sequence T cell receptors that are reactive to MOG from these mouse models that they're using. And they sort of get this little collection of T cell receptors. So then when they treat the mice with the CAR T cells, they can sort of track like which T cells are still there and which ones are not. And so what they did was they just kind of looked at generic diversity of the T cells that were there. So when they treat with their original CAR T cell that they had that's only targeting these high affinity clones or high affinity T cells, versus when they treat with their improved CAR T cell that can deplete both. So they're finding that when they use the original one, only depleting the high T cell one, there's not a lot of change in what's called the effector T cells, which these are T cells that they've seen their target, they've become activated, and they're producing molecules, they're killing things. That's kind of what they're doing. They're in that activated state. Versus when they treat them with their improved CAR T cell therapy, they see that there's a large change in the repertoire where a lot of these T cells are being depleted. So that was kind of their difference that they're seeing, and sort of their explanation for why they see the difference in the results in terms of treating with the CAR T cells, is they're saying, oh, like the enhanced CAR T cell that we made, or better optimized one, is more effective because it's actually depleting those effector T cells that are of lower affinity that they think are really helping to drive disease a lot, versus the ones that are only depleting these high affinity T cells. That's kind of not getting the whole picture. But they still think those high affinity T cells are important because they do find them present in the brain of these mice. And they do find that the mice that have higher clinical scores, meaning more symptoms after the CAR T cell treatment, tend to have more of these high affinity T cells that were not perfectly depleted by the CAR T cells. So that's kind of where they developed this hypothesis of, they think these high affinity ones are important for triggering disease, but both the high affinity and even some more of these low affinity ones are important for kind of keeping the disease going once it starts.
I guess I was kind of wondering, like, is there a risk of over treating? If you go after these T cells like too aggressively, are you going to cause problems? Like, did do these mice become like immune compromised and then they like aren't able to respond to infection, or?
Okay, that's actually a really great question, because this was kind of an important point that I forgot to bring up earlier. But one of the reasons why CAR T cell is of interest is because of its specificity to only targeting these T cells that are driving disease. Because one of the big problems with a lot of the therapies for autoimmune disease that we have now is they're just generically suppressing the immune system. Like they're either stopping the production of T cells or they're producing like these generic signaling molecules that have all kinds of effects on the immune system. So a big side effect and a big issue is people being immune compromised. Whereas with the CAR T cells, the idea is that they're going in and they're very specifically depleting only the cells that are driving disease and that have this particular specificity. So that's kind of their advantage and one of the reasons why they're interested in it. However, there have been like other side effects and different kinds of complications that can occur from CAR T cell therapy, mostly from what we're seeing with the cancer patients that are being treated with these, is there is the risk of like certain neurotoxicity from occurring, like damage to the central nervous system that occurs. I'm not entirely sure why that happens, but it is like a fairly significant symptom that's been reported, though it seems to be temporary and reversible. So that's kind of good, but it is still like a serious issue of concern. Another issue that I know has been reported is cytokine storms, which I don't know if you've heard of these from like COVID, but basically what happens is when you're putting all of these like super activated T cells into someone, it's just causing like this massive flood of signaling molecules to be released and causes this very widespread systemic inflammation that can be problematic, which again, I think that is sort of a temporary thing surrounding treatment, but it is still something that's very severe and that has to be like treated with hospitalization. So those are kind of two of the current risks. And then in terms of long-term, something that's come up that's a little bit controversial, which I don't know, people might disagree on it, is there is some thought that having these CAR T cells in your body long-term might put you at risk for certain types of cancers. But again, I think that's something that's people are kind of debating how big the risk really is and what the cause of that risk is. So that's not really been cleared out yet, but those are just some of the possible like different kinds of side effects you might see from a CAR T cell compared to the current therapies that we have.
You mentioned earlier talking about adaptive. There's some immune cells that are adaptive and our bodies adapt to things, and kind of bouncing off of what you just answered, right, about the body having a really bad time reacting to this influx of signaling. Is there any point where the body could start mimicking the new cells that are being put in the body, or like could the immune system just be like, oh, we actually only like these signals now and we won't accept any other type of disease signaling or something like that rather?
Let me see if I'm understanding your question correctly. So I think kind of what you're asking is if you're flooding the body with these T cells that are specific to let's say depleting like the autoreactive T cells or the MOG-specific T cells in a patient, is that inhibiting the immune system's ability to then fight like germs or something if they get like an infection and those cells have to expand? Is that kind of what you're getting at?
Yes. I'm wondering if at any point, like if this became a long-term treatment, would the fighting cells be unable to identify like other foreign stuff to attack? And would the body itself start producing these cells possibly with like long-term treatment?
Okay, that's a really interesting question. So from what I know regarding cancer treatments or cancer patients that are being treated with CAR T cells, I think, so the CAR T cells go in, they expand, they do their thing, killing the B cells and the tumor cells. But I think once a lot of like the killing is done, they kind of go into a more dormant state and they contract a little bit. So they're still there, but they're not actively killing disease. And it's kind of more of like a smaller background sort of effect that's happening where, so like in the case of these cancer patients where their B cells are being depleted. So there is like a massive rush when, you know, your body's full of B cells and these cells are going after them and killing them. But I think it slows down and becomes more quiet in the background where if it sees like an errant B cell that comes from somewhere, it'll get depleted. But it's not like a really ramped up active immune response. So I think in that case, I'm not aware of any known immune deficiencies in patients long term, like they're impaired to fight different infections, because I don't think it necessarily disrupts the ability of like other T cells to be expanding and mounting responses to like, let's say, a virus or something that they get. So I don't think so. I'm not 100% confident though whether that happens or not. What was the second aspect of your question?
Could the body start replicating the produce T cells that are put in the body?
Your body's T cells that are already there wouldn't produce CAR T cells, because the T cells are like randomly generated, so they wouldn't have a receptor that's similar to the engineered one. Those are kind of different and artificial and aren't there. So it's like you're taking T cells from the patient, engineering them, but it's engineered in such a way that the body couldn't replicate that.
Can those T cells replicate? Can the CAR T cells replicate?
Yes, the CAR T cells can replicate, and generally they will as soon as you administer them and they start seeing their target. That's going to trigger them to cause like a big expansion.
But yeah, like in general, your body's T cells naturally, they expand when you see an infection. And then in most cases, they contract again. And so like you do have a little reserve of T cells that are specific to whatever infection you just fought off. But there are ways that the body like basically tells them like, okay, you can calm down now. And we only need a few of you to like, they're actually called like memory T cells. We only need a few of you to remember what we saw. But we don't need all of you because that will be destructive.
What is this similar to like a vaccine?
Yeah, B cells do the same. So vaccines typically are like causing you to have antibodies, which are produced by B cells. And they do the same thing. They have like memory cells that hang around. So like, yeah, the B cells in your body like form this pocket of memory cells when they see a vaccine like against the flu or COVID or whatever. And then when you see the real thing, they can activate immediately and prevent you from even ever having symptoms because they can just clear out whatever the invading pathogen was.
Could this be developed like a sort of vaccine therapy for folks who are experiencing MS?
You're kind of asking, like, could you kind of give these preemptively as like a preventative?
Yeah.
I mean, kind of like I had mentioned with type 1 diabetes, I think like you definitely could administer them like pre-onset of a disease. I think the problem with that is just identifying when that would be appropriate to give them to someone preventatively. Because CAR T is a very intensive process where like someone has to be like hospitalized for several days, and it costs like, I don't know, a couple $1 million or something for like a patient to get CAR T cell therapy. So we're kind of limited on that aspect of like, they're really reserved for people who are in certain circumstances where the other drugs that we have available don't work for them. But it could be in the future that when we get to a point that they are more readily accessible, we could administer them preventatively. I just think you would need good cause or reason to suspect someone's going to develop a disease before it would be worthwhile to give it to them, because they aren't without side effects. And especially now, because it is a more recent therapy that wasn't started being used until about 2010, like we don't really know what the consequence is of having these in your body long term for the rest of your life. Sure. So I think we kind of need to understand like those questions, because the long-term studies are in process now, and they're starting to like learn stuff 15 years later after people have had them. But I think we really need to understand the long-term consequence a little bit more before we would consider them as like a preventative.
Okay, cool.
I'm just imagining a future where like, in addition to all the regular vaccines, like babies get this broad panel of like protective against cancer and autoimmunity, like CAR T cell type vaccines. That would be kind of a cool future to live in.
That would be. I was just kind of thinking that too. Like, oh, they just give you a little concoction of T cells that can see all of these different things and control all these different processes, and you're good to go. So yeah, maybe a cell-based vaccine is in the future.
I was gonna say I'm quite impressed. Like when I was reading about like the lethality of the MHC CAR T cells, like in how responsive it was. It seemed like not immediately, but like within the week, I think it was like a really fast turnaround of seeing like the mice be affected. So for just like thinking about it, like for my mom, if that was available, like how much pain she would not have been in if she, instead of going to the ER or like even like as part of her regular treatment, instead of having to treat pain, like just being able to treat her body and not like have that super reaction all the time. I think even if it, doesn't wipe it out completely, but greatly mitigates it, but make living with these autoimmune disorders just, more livable.
I 100% agree with you on that. And I think one of the problems that we have too with treating these diseases, because I have Crohn's disease, which is an autoimmune disease of the digestive tract. I was diagnosed in, 2016, I think, at this point. So even just seeing how the landscape has changed is incredible, because there were really 3 biologics available when I was first starting them. But now there's 8 or 9, maybe even 10 that are available. But kind of one of the limitations is we have no way of predicting which biologic is going to be effective for somebody.
Mhm.
So kind of currently you just have to go through this long trial and error process of trying these different drugs, waiting 6 months. Did it do something? And then, if that one didn't work, now I have to pick another one and go through the same thing, wait 6 months, see if it works. So I think the more information we can get and really understand what's happening in these diseases, you can pair someone with a treatment that you have a higher confidence it's going to work. So like you said, it makes a big difference really minimizing the amount of time that people spend experiencing their symptoms before they can find a treatment that works. So if you can just know this person has T cells reacting to MOG and then pop some CAR T cells in them, that would be really beneficial from the patient aspect.
And I mean, just speaking from a legal perspective as well, when you look at the different biologics that exist on the market, all of this also just goes back to how these companies that are developing these different therapies are getting almost 15-year patents on this stuff. And so when it comes to treating people who may be suffering from these conditions, it's maybe this product doesn't even exist on the market for them to even use yet. And maybe we have this basic form while we're still waiting on more generics to get on the market, or while we're waiting on a new discovery, basically, because it feels like a lot of the stuff like this only comes to the general population when prices and economics are called into question a little bit.
Mm-hmm. Yeah, true.
And sort of another interesting point that you kind of reminded me of there is, I remember when we had our lecture on inflammatory bowel disease in class, something interesting that we were talking about was some of these drugs, because I mentioned how adalimumab is only effective in 50% of patients. They're finding that some of the new biologics coming out are effective in only 10% of patients or even less. So just based on the statistics, they're considered failing the trial because they're not working in enough people. But now they're kind of starting to reinvestigate some of those drugs, because for the 10% of people that it works in, it's phenomenal. And it's changing their life, revolutionary that this drug works. But then we kind of don't understand why it works. So we're kind of going backwards and why are these patients different and it works for them when the rest of them don't? So I think that's kind of another interesting aspect too, is it's changing. Like you said, kind of there's the economic play in it, but also maybe the way that we do clinical trials and we sort of evaluate these drugs for effectiveness is something that we should consider too.
And also the way that we categorize patients. I have actually been, now in the last couple of months, I've been to 2 presentations, lectures, talks about using AI and deep learning methods to look back at old clinical trials that have all this patient data and try to see if there's some more relevant sub classification of patients to better understand which ones are responding to different therapies differently. The first one I went to was a anesthesiologist who works in the ICU, and she was talking about different categorizations for the respiratory failure that you get in the ICU. And there's a lot of trials that have failed to, identify a efficient treatment for respiratory failure in the ICU. But going back and using a lot of these new deep learning and statistical methods, they could actually find there might be 2 populations here, and one of them wasn't responding, but the other one really was. And then the second one was actually in this pain conference. It was patients with low back pain, and using the same kinds of approaches to identify patients with low back pain that might be responders to different medications versus others, and sort of doing this more detailed subcategorization. So I think, the development of new treatments plus the development of these ways to categorize patients and better understand the nuances, plus how all of that is going to be understood from the legal side of things, is going to be a very interesting thing to watch over the next few years.
And this is kind of I guess a more field of study, like specific question, but it's like in every field that we study, like the line for like what is effective and what's not effective has like moved like drastically. In like humanities and the law, and science especially, it's like what's effective now, and also like who does this benefit? And so can it almost get like too nuanced? Or I guess I'm kind of wondering about what you were saying earlier about how it like benefits like only, or it like is phenomenal in like 10% of the population. Is that because it causes like more harm to like the other 90% than good, or is it just like not effective altogether?
Yeah, I guess in those instances, it just wasn't effective or it didn't work for the other group. Not that they had like adverse side effects or something that would be considered like a risk for them. But yeah, that's kind of an interesting question though, in terms of moving in the direction of personalized medicine, is just seeing how things are getting so complicated so quickly, or the potential for sort of like the field of diagnosing and treating becoming like very rapidly complicated compared to what it is now. So yeah, that is kind of interesting, because even with CAR T cell, we can kind of see it already in terms of how expensive it is and how laborious it is and what it actually takes to make that effective for somebody. So I imagine that the more complex these treatments are getting, the more of like a financial burden it's going to be, and the more just complicated it is in general to actually get an effective treatment to somebody. So that's sort of something you have to think about too. Is not just like, oh, we have all this cool knowledge and now we can diagnose and treat or whatever, but how does this actually work in a practical sense?
And I think kind of like bouncing off of what Jamie was saying, and like you were saying with economics, is as like part of this, I think part of this revolution with science is the classification on what is considered a person has drastically changed within the last 50 years. Like even just looking at like the civil rights movement of like how Black people are considered people, drastically affects how we're looking at science, right? Like if you're doing a science and your baseline is always a white cis het man, everyone who's not that now, like you said, when you're going back and looking at these studies, it's like, who are these actual populations of like how is this affecting women, and then what kind of woman is it affecting, and then like where are they existing in the world, especially when you have such deep economic disparities of like Flint, Michigan, where they still don't have clean water, or like here in St. Louis, where like they did radioactive testing for like the Hiroshima bomb, and that is like there's still radiation fallout to this day. And so when you're looking at like different populations in different cities, it doesn't make things easier, being like, how do we make medicines that affect a general population when the idea of the general population is expanding and it isn't just considered your default cishet white guy anymore, and that's a really hard thing that science is having to contend with, and in some ways it's easier than others, and also there's also just sweet old capitalism of like how much of this gets to be practical and then how much of it is going to go to shareholders, when like literally insulin is right there as an example. So but circling back to the paper, I would really love Clarice to hear more about like what are your, because you've explained the paper to us I think exceptionally well, and I'm excited to now have more knowledge to spew at random people. But what are your like hopes in getting to do this research, or like what are you looking for to be like your particular part of this project, or like what are you specifically looking for to do with all this?
So some of what I'm working on right now is I'm interested in this rarer population kind of in the autoimmune CNS demyelinating diseases umbrella. So focusing on this disease called MOG antibody disease, or MOGAD for short. And so MOGAD used to kind of be in the mix of multiple sclerosis and another disease called neuromyelitis optica spectrum disorder. And then it wasn't until recently that we really started to, do more screening and just do more testing. And we realized that, there's this group of people that tend to have optic neuritis and more spinal cord symptoms that have these MOG antibodies in their serum. And that kind of led to the classification as MOGAD as a different disease entity from these. And kind of like how I touched on before, like we really don't know what the targets of the immune cells are in these diseases. So with MOGAD, we can guess that MOG is probably a target because these patients have antibodies to it. But in terms of the T cell response, no one's actually shown that or really knows. So that's sort of part of the project that I'm interested in working on, is actually sequencing the receptors of these T cells and identifying how many of them or what's the abundance of them that are reactive to MOG. Because again, no one's actually shown that. So it'd be kind of interesting to know what's going on. And then another kind of interesting aspect of it too is just comparing the T cell receptors across patients to see, like, are they tending to develop similar ones or are they completely different? And sort of what's the abundance? Like, are some people reacting more strongly to MOG than others, and sort of what the deal is there? So that's kind of what I'm interested in, is a little bit of basic science that I think can help kind of push the CAR T cell aspect. And then sort of the second aspect of the project too is we are interested in developing CAR T cells for a particular risk allele that binds a MOG peptide that we think could be relevant to MOGAD patients if they're having these T cell responses against MOG. So that's kind of still specifically like what I'm interested in working on. But yeah, I guess kind of the broader question is just, what are the actual targets of the immune cells in these autoimmune diseases? That's really what I think is the most fascinating question that we really should have the answer to, but we just don't because of limitations in studies, resources, reagents, and everything so far.
Yeah, throwing that excellent question back to Andrew and Knight, what are some things that you're excited to see in the field, or like open questions that you still have like for this field going forward?
I think that overall, I'm just like very excited to see the continued like dedication of research towards studying autoimmune conditions. I mean, I remember a couple months ago, NPR was doing a special for like autoimmune like awareness week, and they were talking to this woman who like wasn't properly diagnosed for 15 plus years, and it like caused her like, I mean, just tremendous like difficulties in life and like tremendous pain. And overall, like I feel like it's one of the like conditions that you can have that's like near the top when it comes to like affecting your quality of life and affecting like what you're able to do and what you're not able to do. And so I think that overall I'm just like excited that there is more of like a possibility to reduce like the effects that these like conditions may have. And I feel like every like line of research starts like somewhere. And I mean, it's amazing how far we've come with cancer research in the last half century, or even less than that, actually. And so I think I'm just overall excited to see where autoimmune research goes from here, because this feels like a big stepping stone.
Jumping off of what Andrew said, like I am also deeply excited to just see more autoimmune research, especially now that we have, at least I hope, being more in the popular conversation about like invisible disabilities, on how like just because someone doesn't look disabled, right, doesn't mean they are disabled. I think I'm also just really excited to hear more about, like, Clarice, what your specific topic is of like what specifically are the cells that are doing this, and especially being able to generalize it between different populations. Like if you can say like, oh, there's these 4 main groups, like we have an actual idea or spread. And I think that'd be really nice to just be able to help name, and something is so scary because it's so vague and personal all the time, of being like, kind of once you get the diagnosis, that's great. But also it's like, having this individualized plan for each person is exhausting. And like you said, it's really expensive. So I'm really excited to see like what finding those broader strokes between people's that is able to connect them, and then, actually being able to treat them. Because it seems like already this has such a positive trajectory for this specific, you mentioned the specific MOG cells that this works with. So I'm really excited to see more of your work and more of your lab and sequencing more and different types of specifics, autoimmunes to help treat, so that we can learn more and just have a proper noun to it all.
Yeah. I mean, I would think I was saying pre-recording that like the whole concept of CAR T cells, when I learned about them in med school in the context of cancer, like totally blew my mind. So this is just one of the like most cutting edge fields of research that I can think of. And I think that it's going to be a really promising avenue.
A question that I had for you guys, kind of like before you came into this podcast, is what did you know about autoimmune disease before you were presented with this paper?
Yeah, for me, I'll say so. Part of my grounding, like I said earlier, was with family. Like my bio mom, has fibro and lupus, and then my brother and my grandmother have asthma, and I think asthma is low key considered to be an autoimmune disease. I used to be a bit more familiar with like autoimmune research as like, because of my mom, so I was more in tune with it. But I'm not as caught up on it within the last, I would say, decade or so. Yeah, about the last decade. So this was pretty new, pretty fresh for me, I would say.
I think that I was not really familiar at all coming in, really. Like I think that I like had some like general knowledge of like how it worked and like what it involved, but I think this paper was a real like eye opener about how it actually works and like how we can effectively treat it.
Cool. I just always like to ask because I think like already 3 out of the 4 of us in this chat have like a direct personal connection to autoimmune disease. And I think it's something that's so prevalent, like just in society in general nowadays with like incidence increasing. But the amount of like awareness and funding that's really being put into the research, I think is still just a little bit lacking. So that's something that I hope to see in the future as well, is just more widespread knowledge about autoimmune disease in general and how is it affecting individuals. Because like Knight, you had mentioned, it is like an invisible disability. So it is very hard. You can't just like look at someone and know. So it is really interesting. And I hope that more people are aware of what it's actually like for these people living with autoimmune disease and why this type of research is so important to be funding, to get these treatments so people can live healthy and not have to live in pain and suffer from whatever symptoms that they have.
Yeah, absolutely. Well, thank you all so much for joining us for this podcast.
Of course, thank you for inviting me. I really appreciate it. I had a fun time.
To wrap up, we have been discussing the paper Antigen-Specific Depletion of CD4 positive T cells by CAR T cells reveals distinct roles of higher and lower affinity TCRs during autoimmunity by J.U. Yi et al. And our expert for this episode has been Clarice Pavlak, and our 2 guests have been Andrew Holder and Johari Hunt. As always, you can download the paper and find previous episodes on our website at inplainenglishpod.org. And if you want to join us for the live show with the Community for Rigor next week on June 12th, you can find the link to sign up in the description for this podcast or on the podcast website. Our logo is by the wonderful team at Biographics. That's Jessica Higginbotham, Tania Lintz, and Yolanda Campos. Our music is by Sam Brunwasser. You can find more of his work on SoundCloud at Visuals Now Beats. And finally, be sure to follow us on Twitter, Facebook, and Instagram at Plain English Sci so that you never miss an episode or exciting news and updates about In Plain English. Again, that's PlainEnglishSci. Thanks once again for listening, and come back next time for another episode of In Plain English.